Okay, hello and welcome to week 2. Thank you guys for hanging with me after the week 1 refresher course. So this week we're going to be getting into a little bit more meaty topics. In chapter 8 we're going to start talking about chromosome mutations, and then in chapter 3 we'll go back and we'll talk about Mendelian genetics. And the reason why I put them in this order is because first we're going to talk about things at the chromosomal level, problems that can occur. And then we're going to go back and talk about inheritance at the single gene level. Okay, so if you remember during meiosis, sometimes things can go wrong, and we call this nondisjunction. What this means is that when these chromosomes are supposed to be being pulled apart, it happens incorrectly and it just doesn't work out quite right. So there's two different places or phases that which you can get this nondisjunction happening. It can happen during the first meiotic division. So you'll have your chromosome pairs aligned at the center of the cell during metaphase 1. However, during anaphase 1, both of the pairs of the chromosomes go to one side of the cell, and then the other ends up with nothing from that particular chromosome. After the second meiotic division, they are now split like you would expect. However, in these resulting gametes, now you have two copies of this particular chromosome from, say, the dad, and in this particular gametes you wouldn't have any. So when this combined with the gametes from, say, the mother, in a cell like this you would have 3 copies. So we call that a trisomy. In this particular one you'd also have a trisomy. Here you'd only have the 1 copy from the mom, and so that would be monosomy. You can also have nondisjunction occurring during meiosis too. So maybe, okay, meiosis 1 happens, during metaphase 1 they line up, during anaphase 1 they get pulled apart appropriately. However, during the second meiotic division, where you would expect to separate these two sister chromatids into the 2 separate cells, they both go to one of the cells. So in this case, half of the offspring, the gametes might be formed perfectly fine. And this is the case when, the normal disjunction happened at the first step. The second step, though, if it gets messed up here, you still can end up with gametes that would have an extra copy or 1 less copy than they should. So when they get combined with the gametes from the other parent, some of the offspring might be completely normal because they came from this line here, these gametes. Otherwise, it could be a trisomy or a monosomy, just like we talked about previously. So when we're talking about variations in chromosome number, there are specific terms that we will use. Aneuploidy, I can colloquially use this as incorrect number of chromosomes, but what we're really talking about here is anything, so we talk about N is the number of chromosomes. In a normal human, we're going to be diploids, we'll have 2N, okay? And we're talking about plus or minus a certain number of chromosomes. In monosomy, you're going to have 1 short of the pairs of chromosomes. In a trisomy, one of the, it's supposed to be all pairs, one of them is going to have a set of 3 chromosomes instead. Okay, so euploidy is a term for, I would say, correct number of chromosomes. Their technical definition, though, is multiples of N. So in most animal cells, we're going to be all diploid, and that is going to be, again, 2N. There are cases where you may have more than 2 sets of chromosomes. This is typically mostly only going to happen in plants, but it can happen in some animal species, like fish, I think, and amphibians, and a few others. So, but polyploidy is going to be where you have more than 2 sets of chromosomes, so you have 3 copies, or 4 copies, or 5 copies of each of the sets of the chromosomes. So if something is triploid, it's 3N, tetraploid, 4N, pentaploid, 5N, and you can go even higher than that.